Good morning students. Welcome to Leela's tutorial. So today let us discuss about development of livestock genomics. So in last class we have seen how the livestock can be improved or the cattle. What is the techniques involved like IVF technique, cloning techniques, all these reproductive strategies are implied in order to make the farm more efficient. So here let us see, after you have the sufficient amount of livestock, you need to develop their genomics. That means the genetically animals has to be developed or improved. So here the development means to improve the production efficiency of the cattle for many decades. So here the livestock development means it is the increasing the efficiency of that particular animal. Though you have gone for the in vitro fertilization or the animal cloning technique, here the efficiency has to be permanent or it has to retain for decade or more than 10 years. Right? If it is only for the particular generations, it is loss for the farm. So here, in order to improve your livestock for the decades, you need to go for the evaluation of the cattle present in the farm. Next one is, it is the evaluation for the development of the animal genetic merit. Once you get the complete farm animals or the livestock, you have to give them the ranking based on their progress, right? Whether they are efficient in increased reproduction, increased milk production, wool production or any other characters, right? So here you are going to rank the animal based on their superior qualities. Right? So here these superior qualities, how you are going to know? By simply taking the biological sample from an animal. So your biological sample can be blood, serum, plasma or amniotic fluid. Right? So like that you are going to collect the sample from the animals and extract the DNA. So next step is the DNA extraction, where you are going to extract or purify the DNA and go for the PCR technique, polymerase chain reaction technique, so that you have the increased number of copies of the DNA. So once the DNA is obtained, you are going to go for the gene typing. So here in the gene typing, you will know what are the complete number of genes present in the chromosomes, the particular chromosomes. What is the gene arrangement? What is the number of genes present? What are responsible for the genotypic or the phenotypic characters which are expressed, right? So like that you will have complete picture of the genes. Then subsequently you go for the genomic evaluation. Genomic evaluation means the complete set of genes can be evaluated once you have the gene typing of complete DNA. Right? So now you have the complete note of the genes which are responsible for the superior quality of the animal and also what are responsible for the inferior quality of the animal. That means you have complete genetic picture of that particular animal. So now let's see, based on the genomics, how we are going to differentiate the animals. So based on the genomics, you can trace what is the functional mutation occurring in that particular gene which is responsible for the variation or seeing the physical or the genetic changes in that group of animals. Right? So here genomics, it offers a unique opportunity to trace functional mutation that contribute to the genetic variation between animals. So here you can identify that particular gene which is responsible for the variation, right? So because of the mutation which has occurred in that particular gene, the differences in the animals has occurred. So based on that also, whether the mutated animal is beneficial or the natural wild type is beneficial, again you will check for the superior quality and get and rank that animal, right, based on the quality. Then next is the organism genes. So again these genes, each and every gene will imply the biological character, the phenotypic character or the appearance, right? So these provide a biological blueprint for its appearance, function and survival. So when you go for this complete chart, the gene typing, if that particular gene is present in those group of animals, then all the groups will have the same appearance or function or the survival, right? So each and every particular gene can act as a blueprint. If that particular gene is present in complete livestock, then all the cattle present in the livestock will have the same appearance, right? Appearance or function or survival rate specifically. Next, it is the genomic evaluation. So here, once you get the complete picture of an animal genetics, here it is a critical factor influencing the animal production and health. The overall chart consisting of the genes, when you evaluate all the animals present in the livestock, then you can conclude which animal is better and you can give them the ranking or the genetic merit to that particular animal. So here that ranking is given mainly based on the EBV. So it is a most important technique which is also referred as estimated breeding value technique. So here in case of estimated breeding value, we check for the birth weight, calving rate, fertility rate, carcasses, etc. And based on that, we will be giving the ranking or the merit to that particular animal. If all the animals satisfy this, then you can go for the further progression. Otherwise, you have to develop the livestock in such a way, separate those animals with superior quality and the inferior quality, then go for the development of your livestock, right? So here, not only livestock development, but also based on their genomics, you are going to develop the livestock. Is that clear? If you have any doubts, put it in comments. Please do subscribe for further videos.